Good afternoon, everyone. This is BP34 Sanika Phatak, and I'm going to present the paper on proteomics reveal the molecular basis of efficacy of human recombinant nerve growth factor treatment for neurotrophic keratopathy. So, a simple background of the author. The main three authors are Damiana Pieragostino, Manuel Lanzini, and Iara Cicalini, and their respective information can be found below. So, the background of the literature was done. So, I will be talking about the background of the literature. A rare degenerative condition of the cornea known as neurotrophic keratopathy is characterized by a decrease or loss of corneal sensitivity, which can result in corneal ulcers, persistent epithelial defects, and epithelial breakdown, damage to the trigeminal nerve, which can arise from a number of conditions, including diabetes, herpes simplex, and zoster infections, neurosurgical procedures, and prolonged use of contact lenses are the primary causes of the disease. So this neurotrophic keratopathy, that is the NK disease, can be divided into major three phases according to how severe the corneal damage is. So the stage 1 is marked by the irregularities in the epithelium, and stage 2 is the consistent epithelial defects. So the epithelial lining of the cornea goes on degrading as the time goes on. And stage 3 causes melting, perforation, and even corneal ulcerations. So traditional treatment for NK, such as artificial tears and serum eye drops, do not address the underlying neural deficiency. Human recombinant nerve growth factor, that is RHNGF, has revolutionized treatment by promoting corneal healing and nerve regeneration. This study analyzes the molecular mechanisms of the RHNGF treatment by analyzing proteomic changes in tear film of NK patients. The researchers of this paper aim to identify biomarkers and therapeutic response, as well as uncover potential new targets for investigation. So this study or this paper gives a detailed proteomic analysis of tear samples before and after RHNGF treatment in NK patients. And the research aims to provide personalized and effective therapeutic strategies, paving the way for more effective treatments. So the introduction, this study focuses on neurotrophic keratopathy, as we have already seen, and it is one of the degenerative corneal diseases corresponding to the reduced or absent corneal sensitivity. This form of corneal epithelial breakdown commonly results in chronic epithelial defects and corneal ulcers. So this study basically raises research questions which are listed below. They are like, what are the molecular repercussions of RHNGF treatments with patients in NK? Can proteomics of tear fluid identify relevant biomarkers and pathways for predicting the therapeutic response in NK? Which proteins or pathways are those that during RHNGF treatment become upregulated or downregulated? This study also shares a few objectives, which are to investigate the proteomic changes in the tear fluid of NK patients before and after the treatment, to identify potential biomarkers indicative of therapeutic response, and to understand the molecular pathways involved in the response to RHNGF treatment. This study utilizes label-free proteomic approach to comprehensively analyze the tear proteome at different stages of the treatment. So methodology, moving towards methodology, the study employed various advanced techniques in proteomics and bioinformatics to analyze tear samples from neurotrophic keratitis patients with recombinant human nerve growth factor. So the sample was collected from NK patients at 3 time points, which is the baseline time, after 4 weeks of treatment and after 8 weeks of treatment. Later, the proteomic analysis was done. It was done by using various state-of-the-art technologies like label-free quantitative proteomics, high-resolution mass spectroscopy coupled with reverse phase chromatography was used. Later, liquid chromatography tandem mass spectroscopy was carried out to identify and quantify the proteins obtained in the teardrops. And the protein identification and quantification was done with at least one unique peptide and intensity-based absolute quantification of abundance measurement. Bioinformatics and functional analysis were also used. The differential expression analysis was carried out by using t-test, which identified the differentially regulated proteins and can be visualized in a volcano plot. The functional enrichment, the pathway analysis for mapping modulated proteins and functional annotations was done. Along with this, also carried out along with various statistical analysis tests like Kruskal-Wallis test and Spearman's non-parametric correlation test. The new techniques and instrumentations which have been introduced in the study are emphasized below. The study on NK patients used label-free proteomics approach to analyze the tear proteome. As we know, the tear samples were collected at baseline after 4 weeks of treatment and after 8 weeks of treatment. Shotgun proteomics was used to identify and quantify the proteins, while in vivo confocal microscopy was used to analyze corneal nerve fiber morphology. The study also characterized and quantified extracellular vesicles in the tear, focusing on those carrying specific markers like CD171, CD126, and CD45. The methodology used innovative techniques like label-free proteomics for unbiased analysis and detailed extracellular vesicle analysis to understand cellular communication and inflammatory processes. High-resolution mass spectroscopy analyzers and reverse phase chromatography techniques improve protein identification and quantification and increase the accuracy. In vivo confocal microscopy provided high-resolution images of corneal nerve fibers correlating with proteomic data. The study also utilized new techniques and advanced instrumentation to enhance the research's depth and robustness. Label-free proteomics allowed a comprehensive teoproteome analysis, while high-resolution mass spectroscopy and chromatographic techniques improved the identification of the new proteins in the sample. The confocal microscopy was used in vivo and provided high-resolution images of the nerve fibers of the cornea, allowing detailed assessment of the nerve regeneration throughout the treatment. The extracellular vesicle analysis provided insights into inflammatory and neurotrophic processes of NK, highlighting the role of extracellular vesicles as signaling vehicles. These innovative techniques offer potential biomarkers and therapeutic targets for future research. Moving towards the research results which the paper obtained. So with an emphasis on the effects of RH and GF treatment on NK patients, in vivo evaluations and thorough proteomic analysis present the main findings of the study. The proteomic analysis showed notable variations in the expression of tear proteins at different phases. The common and unique proteins found at baseline T0 and 4 weeks T4 and 8 weeks T8 of the RH and GF treatment were depicted, and notably 6 additional unique proteins, including protein nucleic acid, D-glycase DJ1 and nucleobindin 2 were discovered after 8 weeks, whereas neurotrophil elastase ELANE and beta-actin-like protein 2 ACT BL2 remained unique until the 4-week point. The differential expression of proteins at these time points were showed by volcano plots. After 4 weeks of treatment, a functional analysis of tear proteins revealed significant inflammation and neuroregeneration, while the upstream analysis revealed the activation of BDNF and BCAN pathways. The downstream analysis demonstrated deep inflammation and predicted new DNA synthesis. These biochemical results were validated by clinical in vivo assessments, which revealed increased nerve fiber diameter by the end of the treatment and increased nerve fiber length, density, and ramifications during the course of treatment. Validating the molecular effects of RH and GF treatment, the study also showed correlation between proteomic data and neuronal responses. So this image was provided in the paper. So as we can see, in vivo confocal microscopy was done, and in the images C1 and C2, uh, the uh, the nerve fiber can be seen to be healed, and the showed an evident increase in subepithelial nerve fiber density together with the dendritic cells at the basal epithelium. Um, Also, the yellow arrows in the images D1 and D2 show nerve fibers, whereas red triangles show dendritic cells. Moving towards the conclusion, the research effectively showed that the RHMGF treatment causes notable proteomic alterations in NK patients' tears, emphasizing proteins related to neuroregeneration and inflammation. Through the use of label-free proteomics, extracellular vesicle analysis, and in vivo confocal microscopy, the molecular mechanisms underlying the treatment's effectiveness were fully understood. According to the research, RH and GF has potential to treat NK by both modulating inflammatory responses and promoting nerve regeneration. These discoveries hence open various new avenues for investigating target biomarkers and refining the neurotrophic keratopathy treatment approaches. So I have just listed down some key findings which, according to me, are highlighted in the paper. So the first is neuroprotection and inflammation regulation. The RH and GF treatment dramatically altered the tear protein profile in NK patients, elevating proteins such as selin BP1, which facilitates nerve growth and repair, and PARC7, which helps protect nerves. The enhanced immune defense following treatment, there was an increase in the presence of proteins crucial for immune defense, such as WFDC2 and LPO, indicating a more robust immune response. IL-6R activation. The significant increase in IL-6R activation following treatment indicates heightened sensitivity to inflammatory cues. The increase in MMP9, which is a marker of inflammation, confirmed this. The fourth and the last one being the neuronal regeneration. The clinical data showed significant improvements in the nerve length, density, and branching in patients treated with RHNGF, indicating nerve regeneration and recovery. This can also be seen real time through the confocal microscopy used, and this was highlighted in the images C1 and C2. So, to list down a few insights which are taken from the manuscripts, they can be: the study provided insights into how RH-NGF treatment changes the protein composition in the tears of the patients, highlighting proteins that help protect and repair the nerves. It showed that the treatment not only helps in nerve regeneration but also modulates inflammatory responses, balancing repair and defense mechanisms. The study confirmed that the changes seen at the molecular level in tear proteins match the clinical improvements in nerve health, supporting the effectiveness of RH-NGF treatment. The findings open up possibilities for developing new treatments targeting specific proteins and pathways involved in nerve protection and inflammation regulation in NK patients. As every study has some future prospects, I would like to add the two which come to my mind as soon as I read the paper. So the first one being the comparative study with other treatments can be done. Perform comparative studies to evaluate the treatment against other standard treatments for NK. This would involve comparing RH and GF with existing treatments such as artificial tears, autologous serum eye drops, or other growth factors to see which one works the best. This type of study helps determine if the RH and GF offers superior benefits or if it can be used alongside the other treatments to improve the outcomes. Researchers would look at patients' recovery, nerve regeneration, and overall eye health to make these comparisons. Also, three test groups can be created, each of them given different treatments, including the RH and GF treatment, and the tier proteomic analysis can be done, and comparative analysis of the proteins which are found in the tier, and the upregulation and downregulation of the genes and the proteins can be observed. This can be also this. So moving to the second one, the exploration of combination therapies. Explore the potential of combining RH and GF with other neuroprotective or anti-inflammatory agents to enhance therapeutic outcomes. For example, researchers could test RH and GF with medications that reduce inflammations or other treatments that support nerve growth. This could involve initial experiments in animal models to see if combination is safe and effective, followed by clinical trials in patients. The goal is to see if combining treatments can provide better results than using RH and GF alone, potentially speeding up recovery and improving nerve regeneration. Also, I would like to add here that this treatment can be studied on the viability of the treatment, like how long the nerve remains regenerated, or after stopping the treatment completely gradually, the nerve regeneration stops and the nerve again, like the NKDCs again relapses in the patient, or it gives an ultimate solution to the patient's problems. So the here I have attached the manuscript on which the presentation is based. So this manuscript was taken from the Nature journal, and yeah, thank you for giving a patient ear to this presentation. Thank you.